All right, moving on to our next presenter. Benizou Rousso is going to be presenting. I hope I haven't butchered your name, but he's going to be presenting on the application of fluorescent probes to distinguish species compositions of cyanobacteria blooms. Thank you.
Thank you for the introduction. So today I'll be exposing a little bit, talking about the application of fluorescence probes to distinguish species in cyanobacteria blooms. In this presentation, I'll be exposing actually the proposed outline for my PhD at Griffith University. I have started around 5 months ago, and Dr. Eduardo Bertoni and Professor Rodney Stewart are my supervisors. So in this presentation, I'll be talking about the concept, the idea that we're trying to pursue in my PhD, so a little bit of a difference in comparison to the other presentations. But also I'll be presenting some preliminary results that we got so far by historical data analysis, besides some previous lab work that was done in partnership with the Australian Rivers Institute, which Anne Chuang and Professors David Hamilton and Michelle Burford are members of. So the concept of the presentation will be that. So I was lucky enough that Florence and Sarah have already talked a little bit about fluorescence probes, so I can go super briefly and quickly about this. But just to briefly talking about the subject, what are the fluorescence probes and how they work? So basically they work based on the reflectance of light harvesting pigments of algae, such as chlorophyll A, B, C, carotenoids, and phycobiolants. So a specific frequency of light is emitted towards the pigment. This is called the excitation wavelength, and then the pigment absorbs it and reflects it in a higher frequency, which is called the emission wavelength. And this specific trait makes it able to us to identify this sort of pigment in a water sample. In regards to cyanobacteria, the phycocyanin is a pigment of interest since it's unique to this phyla, and it also is the pigment that characterizes and gives the name of blue-green algae, and also has the specific range of excitation and emission, so it makes it possible to identify it on a water sample. So what are the benefits of using fluorescence probes? So some of the benefits is that it can provide real-time measurements and relatively cheap estimatives of total algae and cyanobacteria biomass. However, as also was stressed especially by Florence, there are several interferences that need to be accounted for in order to obtain reliable results from those kind of devices, such as the variation of water temperature, the turbidity, and even some morphological traits from the monitored microorganisms, such as do they form colonies? What is the distribution of the microorganisms in the water sample? What is the cell size of the microorganisms? Despite all of that interferences, this subject has already been studied a lot in the literature, and we know a little bit how to overcome and to correct them. Actually, this figure here was taken from a recent published review paper on the subject, and it shows how some of the variables link or correlate with the phycocyanin, which is measured. Is it possible to see? With the phycocyanin that is measured by the fluorescence probe and the actual phycocyanin in the water sample. So in the figure, the green arrows represent positive relationships between the variables. Red arrows means negative relationships, and blue arrows represent situations in which there is no clear correlation yet or controversial evidence is found in the literature. So given that, we can see that phycocyanin fluorescence can provide a total estimative of total cyanobacteria biomass already. We have plenty of studies that show that. But what about take a further step on and make the distinction in between this total cyanobacteria biomass into species of interest, such as non-toxic or taste and other producers? So in the literature, there is already some previous work that has used fluorescence probes to distinguish in between different algae phyla, and this has been done to distinguish, for example, between green algae from blue-green algae from golden algae, and this is mainly because those phyla have different pigments that have those these specific spectral characteristics, and then make it possible to have different wavelengths to distinguish between them. But then when we try to go towards the differentiation of in the level of species or genera in the same phyla, in other words, trying to use those probes to distinguish in between cyanobacteria species, or at least at the general level, we face a hard challenge because there is no clear spectral difference in between them. So one way to overcome that is to develop models, algorithms, like data-driven models, to estimate the composition of cyanobacteria by combining the fluorescence probes, monitoring data, such as the chlorophyll A and phycocyanin, with historical and current environmental conditions, such as water quality, nutrients, pH, oxygen, and even cyanobacteria monitoring data, such as cell count and biovolume in identification of the species. Those two data sources together can then be used in modeling, using modeling techniques such as principal component analysis or artificial neural networks, Bayesian networks, maximum entropy models, in order to try to find correlations of patterns of appearance of certain species according to scenarios, and then try to develop a model based on the fluorescence probes to distinguish species. So it's within this context that my PhD will be placed, and will have as main objective the development of a real-time cyanobacteria bloom composition model. This research already has as an industry partner SEQ Water, and altogether we have defined the Dingalpa Lake, which is a Queensland drinking source reservoir, as the location where the study will be conducted. In order to achieve the main goal, we have ranked some specific objectives, such as the characterization of the key species in the water body, the calibration of the fluorescence probe, which is in the case is the exosilent probe, and the identification of key environmental triggers to blooms and species dominance for the site of Tingalpa Lake. So talking a little bit about the Lake Tingalpa, it's located at Cabalaba, South Brisbane, and is bounded by the Leslie Harrison Dam, has a total area of 87 square kilometers and maximum full capacity of around 13 megaliters. Its waters are used mainly for treatment and drinking purposes, being recreational activities not allowed at all. And one of the reasons that made us choose this location is because SEQ Water has reported to us a recurrent problem with algae blooms and taste and other events in the raw water, besides having a long historical data set of water quality and cyanobacteria monitoring, which includes, for the more recent years, fluorescence monitoring using a vertical profile sampler. So we have a lot of data that can help us to try to find these patterns. In this figure, I expose the conceptual framework of the PhD. So the model itself would have three components. The first component to the left in green would use historical data from cyanobacteria monitoring, either cell count and biovolume, and water quality data, pH, nutrients, temperature, all of that together. And through data analysis, which is currently being performed, we'll try to identify the key species of the water body and try to find correlations among the other variables in terms of the frequency of appearance and dominance of those species according to environmental conditions. In that way, we expect that we can develop a component, which is a model that will give us a likelihood of cyanobacteria species based on environmental conditions. So this is the first component to the left. The second component to the right in orange will refer actually to the calibration of the probe to the location, and will be responsible to provide us an accurate estimative of total cyanobacteria in the lake. For that, we have to do some lab work that will be based and directed by the results that we achieve in the preliminary analysis that will give us characteristics of the specific site that we'll be dealing with. The third and final component then will be responsible to merge those two components and provide us a real-time likely cyanobacteria species prediction model. So going a little bit towards what we already have seen so far, the preliminary results, just starting giving a brief overview of the data, we have 15 years of monitoring, ranging from 2003 to 2018, with 5 sampling points across the lake. In regards to cell count data, which was the one that was mostly analyzed so far, there were a total of 236 genera monitored, from which 109 were at least once detected. From those 109, 24 genera belonged to the Cyanobacteria phyla, and in terms of order taxa, the Synecocallis were the most abundant in between them. Further on, for data analysis, we selected only the cases when at least 95% of the total Cyanobacteria bloom was discretized in identified genus, so we could have a more reliable data set of species compositions within a bloom. In this graph, we show the frequency of appearance of each genus for 3 scenarios that were defined according to the magnitude of the total Cyanobacteria cell count. So the scenarios are less than 2,000 cells of total Cyanobacteria per milliliter, to the left here, then in between 2,000 to 10,000, and greater than 10,000. One tendency that we can observe, at least preliminarily, is that, I don't know if the color is okay, if with the increase of the total Cyanobacteria cell count, we also have an increase of frequency of appearance of a Phanocapsa, which is the red bar. And the opposite is true for Microcystis. One of the explanations that we have towards this is that a Phanocapsa has a way smaller cell size, so in order to generate the same biovolume of Cyanobacteria, it will require a greater amount of cells. But nevertheless, this is going to be afterwards cross-validated when we analyze the biovolume data. But observations like those can help us to identify patterns in which there's more likelihood, or at least how is the frequency of appearance of certain species of Cyanobacteria according to scenarios, which is the purpose of this component. Another pattern that we could observe is that there is a temporal uniformity of dominant species throughout the reservoir. So we can see this through this time series graph, in which each color represents a different genus, a different species of Cyanobacteria, and each panel from up to the bottom is a different location on the site. So for instance, we can see that in the beginning of 2012, I don't know if it's too big, you guys can see, but here, there is a dominance of a Phanotessi, I don't know if it's pronounced like this, sorry, in yellow throughout the whole reservoir. And the same occurs at the end of 2013, where a Phanocapsa in red also dominated all the blooms, even if the magnitude was different in between the sampling points. This is also true for 2016, when Microcystis in purple had dominated from June, July till the end of December. This shows us that, at least in terms of cells, not in biovolume, and for the sampling points that we have, that the reservoir is well mixed and the dominance is quite homogeneous along the whole area. Another pattern that we can observe throughout this time series graph is that the bloom crash seems to be preceded by the increase of a second or third dominant genus population. So for instance, in the beginning of 2018, we can see that the bloom was mainly dominated by Phanocapsa cells, and then after that there was an increase in the populations of Cyanocatina and Cyanogrannis, and yellow and green. And following that, the total Cyanobacteria cell count started to decrease until total bloom extinction. So this behavior as well can show us and help us to identify patterns and model what is the species that is dominating. Another point that we have observed is the comparison between the historical data set of cell count taken traditionally in the laboratory by microscopy, which is the blue line in the graph, against the outputs of the fluorescence probe currently in Lake Tingalpa that are represented in the gray line and the orange line in the graph. The gray line refers to the raw fluorescence units, RFU, given by the probe, whereas the orange line refers to the built-in conversion that the manufacturers provide in the probe from RFU to cells per milliliter. So the first thing that we can see is that the built-in conversion is not accurate at all. So just buying the probe and put it in the water would probably not give you a lot of reliable results. So as the literature shows, site calibration is definitely needed. Also, we can see that the gray line also really does not fit well the blue line. And this is probably as well because we are not accounting for the local interferences as we should yet. But despite that, one specific trait that is of interest, and we could notice in here, is that usually after the bloom has already crashed, so after the moment where the blue line decreased significantly, the gray line, the RFU, is still high. And one of the hypotheses that we have is that this might be happening because the probe is actually accounting for the pigments that were inside of the dead cells, now are dispersed in the water column. So going towards the lab work that was done previously in partnership with the Australian Rivers Institute, there are some observations that might support this hypothesis. So the lab work experiments have analyzed different multiple cyanobacteria species behavior in different growth stages and conditions. So for instance, here we have a graph to the left of the comparison of the readings of phycocyanin against the growth and the cell density of microcystis. So you can see that with the increase of cell density, the phycocyanin measurements follow quite linearly, but after we had the crash of the population, the phycocyanin decreased but remained higher than before. So this behavior that we observed here may support, or is at least quite similar to what latent alpha is showing. And by last, another point that might be of interest that was analyzed in the lab was the difference of phycocyanin readings by fluorescence when we compared mixed cultures and single species cultures. So in this table, we have 19 different combinations of different proportions of 4 different species. And then we measured the phycocyanin readings for the mixed culture and compared the expected one from the readings that we got from the single species cultures. So what we can see is that at the bottom of the table here is that for the cases where microcystis and cylindropersmosis dominated or comprised the mixed culture, we had the biggest difference in between the phycocyanin readings for the mixed culture against the expected one by the proportional sum of the single species. So what this might indicate for us is that the calibration of the probe should be done considering the key species of a certain reservoir, for instance. So just to wrap it up, the presentation that has a lot of information that is not really tied together, we can see that the data set has a variety of scenarios, and from that we believe that we can achieve and retrieve some interesting results through data analysis. We have already found that despite the presence of several cyanobacteria species, only 3 to 4, while using cell count as reference, were predominant, which is good and for us in this case, and shows us like a simple case for studying to apply this bold, I would say, technique. This will be cross-checked while further investigating biovolume data and seeing if there are only those 3 to 4 predominant species, and then cross-check with environmental conditions. Further, previous lab work done shows the potential of how combining both data analysis and directed lab work can help us to understand what the probe is reading, its limitations, and then give us a light to where we should go in order to overcome these limitations. So by last, I would like to say that this research is still on its early stages, and despite I have presented some preliminary results, and we might try to focus a little bit on that, the main goal of this presentation was to show to you guys the conceptual framework of the PhD and really trigger discussion and see what specialists think about that. So there are some references and acknowledgements, especially to Griffith University and SEQ Water for funding this research. Thank you.
Thanks, Benny. We might sneak in one question if anyone's got one question.
Hi, thanks for the talk. That was interesting. Some of those cyanobacteria you're talking about are very, very small. They have a very low biovolume, so like your Aphaenocaps, your Aphaenothecae.
Yeah.
Have you considered and I imagine they might reflect very similar light because of the size and the shape and so on. Have you considered maybe grouping some of those together, and maybe even something like the Cyanocatina and Cyanogranus, where there's actually iron associated in those colonies? I don't know. Someone else might know more about fluorescence, and but some of those ones where I assume they might produce a different colour again, or a different wavelength, or you might get some interesting results by actually putting those things in groups.
Yeah.
That's just from a taxonomic kind of point of view.
Yeah, that's a good idea, and this is probably going to be done on further stages when we're applying modeling techniques. So there's cluster analysis, principal component analysis, but definitely grouping them is a good way to try to figure it out.
You might find all the cyanobacteria that are less than maybe 20 micrometers. You could put them all into a group together, and then your larger ones, which are your Dolichospermens or your Chrysosporums or whatever they might be, could go in another group, and you might get much more distinct differences in your results. Anyway, just a thought.
Yeah, thank you. Thank you.
We're out of time, so thank Benny again.